Good good afternoon, everybody, and good day, good morning, depending on where you're joining us. Welcome to this session focusing on international multilateral cooperation, specifically talking about the COVID-19 response through BRICS science and technology and innovation collaborations. I'm Zoleka Ncete, a program manager at the South African Medical Research Council. I will be based in Cape Town, South Africa. I will be moderating this session. As you all know, the COVID-19 pandemic caused by the SARS-CoV-2, it continues to be a major global health challenge. BRICS countries, Brazil, Russia, India, China, and South Africa account for more than 27% of the total global COVID-19 burden. And in response to the pandemic, and in the absence of treatments and preventative vaccines at the onset of the disease, the BRICS SDI Framework Program launched a call for multilateral basic, applied, and innovation research projects in an effort to facilitate cooperation among the researchers and institutions in the 5 BRICS countries. And in this session, we will be hearing from some speakers of some of the projects that were chosen to be supported through this initiative. And we selected 12 joint projects for funding, of which 7 of them had a South Africa component. The key questions that our speakers will be addressing is how the projects are responding to the COVID-19 pandemic, how the BRICS countries are working together, and what lessons can we learn for international cooperation in general. We have several speakers now representing 4 projects that we have supported. I will invite the speakers now to switch on their cameras so we can quickly introduce them. We have on the panel, we have Professor Kelly Chibale, who is a full professor of organic chemistry at the University of Cape Town. We have Professor Hanley Smarts, who is an associate professor in the Department of Informatics at the University of Pretoria. We have Professor Anneke Hasling, who is a South African clinician scientist working who is the director of the Desmond Tutu TB Center. And we have Dr. Edwin Wilkinson, who is a senior scientist at the Center for Epidemic Response and Innovation based at the School of Data Science and Computational Thinking at Stellenbosch University. Welcome to the panel, and I look forward to a great session with you today. So just before we start off with our presentations, I just wanted to remind the participants in the session that you can send through your questions through the chat function, and you can use the QR code. And just to note that we will run through the presentations and we will have a question and answer session at the end of all the 4 presentations. We will now start off with our first presentation of the afternoon. Dr. Edwin Wilkinson will be talking about the SARS-CoV-2 network for genomic surveillance in Brazil, Russia, India, China, and South Africa. And I know on your program you have Prof. Tulio de Oliveira, and I would like to extend my apologies on his behalf because he unfortunately was unable to join us this afternoon, but we are in the capable hands of Dr. Edwin Wilkinson. Dr. Wilkinson, over to you. Thank you.
Thank you, and a warm welcome to everybody. Should I start sharing my screen?
Yes, please.
Welcome everybody. I'm Dr. Edwin Wilkinson from Stellenbosch University, and myself and Tulio are part of the Network for Genomic Surveillance in South Africa. So this network consists of a whole bunch of academic institutions throughout South Africa, including the University of Cape Town, Stellenbosch, the University of the Free State, CRISP at UKZN, as well as WITS, the National Institute for Communicable Diseases, and then the University of Pretoria. And this network is funded by the Department of Science and Innovation, as well as the South African Medical Research Council. So my talk today will mostly focus on the identification of the new variant of concern that was identified by our network last week in South Africa. Since then, this new variant has been designated Omicron by the WHO, and according to the pangolin lineage assignment, it's called B.1.1.529. Next strain has also came up with its own classification, which in next clade it will be 21K. Since the initial discovery, 329 Omicron genomes have been made publicly available on the Gizate sequence database, of which 172 are from South Africa. And currently we have sequences from Gauteng province, the KwaZulu-Natal, the Western Cape, the Eastern Cape, as well as Mpumalanga. But multiple sequences have also been identified in other locations throughout the world, including in Europe, North America, and in Asia. There are some signs from laboratories that Omicron is likely to be present in all provinces, and I will revert back to this a little bit later. We can make some predictions currently about the properties of this variant based on the genetic mutations that it has, but more work is urgently needed to fully understand the impact that this variant can have on the pandemic. And currently, vaccines remain the critical tool to prevent severe disease, hospitalization, and death. So currently, this is the epidemic curve for South Africa. We had a first wave in winter 2020. Then we had our second wave, which was dominated by the Beta variant. That was in December, January, and February of 2021. And then we had a very severe third wave during the winter of 2021, which was dominated by the Delta variant of SARS-CoV-2. And as you can see here, South Africa is currently in an epidemic upswing. So this is the data from our world in data from yesterday, but more than 8,000 cases has been identified yesterday. So this needs to be updated. So just looking at the province of Gauteng, these plots basically show the SARS-CoV-2 incidence on a log scale. And you can see that in each of the metropolitan or in each of the municipalities within Gauteng, there is a sustained increase in incidence in the past 2 weeks. Looking at other provinces within South Africa, we're also starting to see the first signs of this sustained increase in other provinces. So looking at the hospitalizations within Gauteng, it does seem that there is a small increase in hospitalizations at the moment, but it's still very early within the fourth wave. So we will have to monitor this a little bit more closely in the weeks ahead. So the big cause for concern with Omicron is its mutational profile. So as you can see here, this is the entire genome of SARS-CoV-2. This highlighted area here is just the spike region of the virus, which primarily interacts with human hosts. So in total, we found 30 mutations within this spike domain, 15 of which occur within this red area called the RBD, or the receptor binding domain. And this is pretty much what's causing most of the concern regarding this variant. In a phylogenetic tree, this variant is distinct from other circulating variants of concern or variants of interest, and is not a direct descendant of either delta or beta, even though it carries some similar mutational profiles or similar mutations. Some of these mutations, the 30+ mutations, are well characterized, and we know the impact which can lead to heightened transmissibility or immune escape, but many others we have not seen before and still needs to be characterized. So their full significance is still uncertain. So I alluded to, there are signs that the variant is present in most of the provinces within South Africa. This is not based on sequencing data. This is based on qPCR data. So in this graph here, you can see the total number of test positive cases by the TACPath assay. You can see the clear first wave, second wave, and third wave. But we've seen in recent weeks that there's a massive dropout in spike using the qPCR test. And this is indicative of Omicron presence. And we see this pattern in all 9 provinces throughout South Africa at the moment. So you can see that it went from virtually less than 5% to almost above 50% nationally. So looking at the variant profile for South Africa over time since the start of the pandemic, the first wave was caused by B.1 variants, which is very similar to the Wuhan reference with the addition of the D614G mutation. Then the second wave was caused by the Beta variant, which was then quickly replaced by the Delta variant as the third wave progressed. These ones in yellow is another variant that we characterized a couple of months ago called C.1.2. But you see here in the past 2-3 weeks that the Omicron, or the B1.1.529, has shot up in prevalence to above 80%. This suggests that the Omicron variant is outcompeting the Delta variant. So looking at the various provinces in South Africa, most of our sequencing capacity nationally is based within the 3 most populous provinces. That's KwaZulu-Natal, Gauteng, and the Western Cape. But we, as a national network, have also reached out to other centers, and we've identified Omicron in the Eastern Cape as well as in Mpumalanga in recent days. So work is currently underway to fully understand the impact of Omicron. This pertains to its transmissibility relative to other variants, its efficacy to vaccine protection, the risk of reinfections, the efficacy of therapeutics, the disease severity, and the effect on diagnostic assays. So just to summarize, epidemiological data suggests sustained increase of COVID-19 incidence, initially across Gauteng, but now across most provinces in South Africa, which suggests that we are entering a 4th wave. It looks like this 4th wave will be caused primarily by the new variant, the Omicron. Omicron is detected in multiple samples, as I mentioned, with this signature qPCR S gene dropout, which suggests that it's already widely spread across the country. The mutational profile and the epidemiological picture suggests that Omicron is able to get around some of our immune protection to cause infection, but the protection against severe disease or death from vaccines should be less affected. Therefore, vaccination still remains the most critical tool to prevent severe disease and death in the weeks ahead, and public health and social measures remain critical to slow its spread. So just in summary, this work is made possible by a really large group of people across South Africa from the various teams. And there's a couple of pictures in the previous slide, but the numbers of people that are really involved on the ground is staggering, and I can't go through all of them at this stage, but they are right here on the screen. This is for all the universities, as well as our colleagues in private labs, as well as international colleagues in Brazil, the United Kingdom, and in other places. Thank you very much.
Thank you very much, Edwin. I feel like we are, I'm feeling quite special today. We are we're at the cutting edge of this development regarding this new variant. We really appreciate you taking through what we currently know about Omicron and the evolution of the pandemic in South Africa. And just before I move to the next speaker, I just want to remind the delegates that you can post your questions through the chat room as we go, but please make sure where possible, if you're asking a question from a specific delegate to a specific panel member, to indicate that in your question so we can direct your question appropriately. We are now going to move on to Professor Kelly Chibale, who is a full professor of organic chemistry at the University of Cape Town, where he holds the Neville Easdale Chair in African-centric drug discovery and development. He's going to talk to us about the multidisciplinary platform based on artificial intelligence for accelerating drug discovery and repurposing for COVID-19. Over to you, Kelly. You can start sharing your slides.
Thank you, Zoleka. Sorry, I don't seem to see my slide appearing on the. My apologies. Carolina, can you share that slide?
Yes, I can share for you. Wait just a minute.
Please do. That will help. Sorry, I seem to be having an issue here. Thank you.
Oh, you're welcome.
And as I go through, Carolina, I'll just say next slide, and then
definitely. Okay.
Fantastic.
So can you see the slides?
Yep, I can see. If you can put it on.
Okay.
Excellent. Thank you very much indeed.
You're welcome.
Thank you, Zoleka. Thank you for the very kind introduction. This is a presentation I'm going to share with my colleague, Professor Carolina Hota Andrade from Brazil, and we are representing our partners in the consortium. If I can have the first slide, please. Next slide. All right. So basically, I'm sharing this presentation with Carolina, and I'm going to cover the first part. Carolina will cover the second part, and I'll come back and finish the last part. So this is a program involving 3 main PIs, myself in South Africa, Carolina from Brazil, and you'll hear from her very shortly, and our colleague, Professor Vadim Makarov from the Russian Academy of Sciences. And we really got together when in response to the call, to formulate and conceptualize this project based on utilizing the power of artificial intelligence to respond to the COVID-19 pandemic from a therapeutics perspective. I think we've heard a lot about vaccines, and I think people need to really appreciate the fact that the role of therapeutics in COVID is going to become very important as well. Not just because of what we just heard about from the previous speaker with new variants, but also the challenge of providing access or equitable access to these tools becomes important. So we need multiple interventions to do that. So this is the consortium, and of course, leveraging on our respective partners. And I've brought in other partners from Brazil and the institutions are indicated here, and a collaborator of ours from the US, Sean Eakins, who was an expert in artificial intelligence. So the goal of this project on the next slide you see here is really twofold. Firstly, is to use artificial intelligence and machine learning models to focus on repurposing drugs that have been approved clinically for use in other disease indications. And this is a strategy to fast track the process so that we can get into clinical trials as soon as possible. But this is driven by the protein targets, which I'll touch on very briefly. The second objective is to really exploit the AI computational technology to look through our respective libraries of molecules that we've assembled over the last number of years and see how we can identify new starting points for COVID-19 drug discovery processes. Next slide, please. So we're going to do now, before Carolina comes in, I've got 2 slides where I'm just going to walk you through these 2 workflows here. They are very busy slides, but I can assure you the message is very, very simple. Firstly, what we're focused on doing, and this is something that Carolina is driving on behalf of the consortium, which she will speak to shortly, is building machine learning models using data that is available from the various antivirus screens, but also data available using our particular targets of interest. In this case, these are the dihydroorotate dehydrogenase, the human dihydroorotate dehydrogenase, DHODH, and proteases that you will see Carolina speak about as well. After that, apart from deploying those machine learning models to search for approved drugs that have been approved by the FDA in the repurposing approach, we're also using these models to do a virtual screening campaign to look at the established chemical libraries that you see under number 2, libraries that have been assembled by our colleagues in Brazil, and libraries that we've assembled in Cape Town, and libraries assembled by our colleague in Russia, and really sieve out what are the potential starting points for drug discovery. And then the third bullet that you see here is that once we identify those potential starting points, we then want to verify the computational predictions and then test those in the assays that are described under 3, which are provided by our colleagues in Brazil. Next slide, please. And continuing from where I left off on 3, so what you saw in 3 on the previous slide was testing molecules that have been computationally predicted to bind to the proteins of interest, which were developed from the artificial intelligence machine learning models, and then tested in a biochemical experimental assay to verify the computational predictions. After that, the next step that you see is then to see whether those compounds are not harmful to normal cells. So we do cytotoxicity screening, and then we focus on those molecules that have a reasonable therapeutic window that are not harming normal cells. And then we basically test them directly on the COVID virus for antiviral activity. And that's what you see under 4, where the arrow is now pointing. And then once we identify anything that is active in terms of killing the COVID-19 virus in this host cell assay, that then represents a starting point for doing chemical modifications to generate derivatives that we can optimize with respect to efficacy and safety, including drug metabolism. In parallel, when it comes to the repurposing approach, where we identify medicines that are already approved for other indications, we're using nanotechnology or nanoscience under 7 to develop particular formulations of those drugs that could then be deployed into the clinic for clinical testing. And of course, one of the expertise that H3D is providing when a drug is identified that is already approved for use is we can do human dose predictions from our drug metabolism platform. So these two slides and this one here really just give you a flavor of the integrated nature of this program, integrating the various aspects from artificial intelligence, machine learning, computational chemistry, to biochemistry, medicinal chemistry, and drug metabolism, as well as nanotechnology. I'm going to hand over now to my colleague, Carolina, to speak to the next slides. Over to you, Carolina.
Okay, thank you so much, Kelly, for your great introduction of our project. And what I'm going to show you today is some of our computational results. So we have been working for more than 8 months, I think, when we started this collaborative project. And we had developed all the computational models, artificial intelligence models that we are now applying those cascades on those virtual screenings to select and to prioritize compounds for testing. So the first model we developed is the machine learning model using data from the literature for SARS-CoV-2 phenotypic data. So how is artificial intelligence changing the drug discovery project? So artificial intelligence has the capacity of accelerate this project, these discoveries, by using data that have been already published on the literature. So we use data, experimental data that have been already tested and published in literature or available in public database such as PubChem, Campbell. So we gather all this data from compounds with biological activity tested. So in this case, the compounds have been tested in the cytopathic effect, phenotypic assay against the SARS-CoV-2. So we used the known actives and inactive compounds, published them, and we started training our models, generating machine learning models. And if these models are robust and predictive with appropriate metrics, so we can use these models to predict new compounds that have never been tested. And with that in hand, we can learn with data that have been already generated, and we can get more potent and more selective compounds to accelerate the discovery of a new drug candidate. So in this case, we generated those models for SARS-CoV-2 phenotypic data using different descriptors and different machine learning models. We ended up with this, the best model with the metrics here. And we moved for our target-based approaches. So in this case, I'm going to show you the proteases sub-projects in our BRICS collaborative work. So as the virus, the SARS-CoV-2 has two protease. So this is the main protease and the papain-like protease. So in this case, we used all protein structures available in the PDB. For the main protease, we analyzed all of the structures, and we generated and validated many kinds of computational models, as we can see here. So we developed and validated docking models using the structures from the PDB. We also developed this kind of ligand-based model called shape-based or pharmacophore-based. So based on known inhibitors from the literature and known inactive compounds, we can validate, statistically validate the model, the computational model. And we also generated machine learning and deep learning models for the MPRO inhibitors. And with all these computational models, we applied them in a cascade, in a virtual screening, using, in this case, the H3D in-house database of synthetic compounds that have been synthesized all over the years. So there is more than 6,000 compounds. And we started filtering out, prioritizing compounds, passing through our validated computational models to select or to prioritize those hits, those virtual compounds that we are going to experimentally validate and test. And our chances of success is much higher than if we are going to test all of them randomly. We did the same for the other protease, so the PL-Pro protease, the papain-like protease. We developed shape-based models. We validated those models when we also developed ensemble docking models using various protein structures from the PDB to try to capture the flexibility of the protein, which is very important in the ligand binding. We validated the models using the enrichment studies, and we also performed the virtual screening to select, to prioritize compounds from those data, from H3D database. And all of those compounds that have been selected, they are being shipped out to Brazil, and we are going to start very soon, hopefully as soon as compounds arrive here, we are going to start the enzymatic assays with the proteases. Just to show you here, this is the crystallographic structure of PL-PRO with this experimentally validated inhibitor, and our compounds were docked exactly in the same BL2 loop. And as we can see here, there is this other region called BL2 groove, which is very hydrophobic, and we can also explore this for the further optimization, hit to lead optimization, as Kelly just mentioned. So we are developing, we develop all the computational models. We have many other results using other databases, and also for drug repurposing. We started testing some drugs in the scenario of drug repurposing, and also in the sub-project of the spike protein. And I think that's it I was going to show you today. And I will get over, the word is now from you again, Kelly. Thank you.
Thank you, Carolina. Thank you very much. Could I please have the next slide, please? So there are just 2 slides remaining. And of course, we were given the remit to also reflect on what's happened so far, and talk about some of the challenges that we have encountered, but also some of the successes. And then there's one more slide that will come after this when I will talk about the lessons that we've learned so far from this program. Some of the challenges, of course, challenges are opportunities. So it's not really, we're not complaining. We're just highlighting some of the things that we've had to navigate to get the collaboration going. And of course, it's really that we're bringing together very different cultures. There's a Russian culture, there's a South African culture, and there's a Brazilian culture. And so it was a bit of a challenge to get the agreements going in different languages. And so that was a major issue. Maybe we should find one BRICS language going forward, but that's for the politicians to sort out. And I'll skip the coordination, that's inevitable. But of course, Carolina was just talking about the compounds that have been identified from our database, from the artificial intelligence machine learning that we have shipped to her lab in Brazil. But you can imagine that the time it's taking now to ship things across borders is a bit of a challenge now with the pandemic. And so that's an obvious challenge that we've had to deal with. And of course, language. Of course, you've heard Carolina speak. She's very fluent, but not everybody is fluent. And actually, our colleague in Russia is also very fluent. But of course, you know, that can be a barrier sometimes with these languages. What about the success? We just got going on this program over the last few months. We have been very proud of the success so far. It's taken quite significant effort to assemble the sub-project teams that are focusing on each of the targets of interest. And we've brought together really a real team with complementary skills, sharing knowledge and data and meeting regularly as the project has kicked off in a very powerful way. And then finally, but not least, we've actually developed a data sharing mechanism, which is allowing us to share things online, and of course, cutting out emails and any other kind of bureaucracy. The final slide, if I can have the next slide, is really the lessons that we've learned from this wonderful collaboration with Carolina and our colleagues in Brazil and Russia, is of course the fact that this is a truly interdisciplinary project. Of course, drug discovery, but by its very nature, is an interdisciplinary project. You need to integrate chemistry, biology, and pharmacology. And that's the only way that you're going to make progress in this. But that partnership is based on complementary expertise. And I think you heard from Carolina's presentation that she brings something unique that we don't have in H3D at this point in time. But we are building that as we learn from Carolina and other partners. And of course, we have When you have a team like this, a group in Russia, a group in Brazil, a group in South Africa, different cultures, it's no longer just about the science. It's about managing the project. So project management becomes a very important component of this kind of partnerships. And I really emphasize to people pursuing projects in academia, you gotta bring in aspects of project management, because when the team gets bigger, the projects get bigger, you can get lost without a very structured project management system to keep everyone focused on the goals. I'll skip the obvious thing about regular meetings. It's been very useful, but also, you know, the one lesson was that it was actually easier to deal with a particular institution. So when we at the University of Cape Town dealt with Carolina's institution on a bilateral level, it was actually easier than trying to deal with all three partners at the same time. So the big lesson is that we should keep the multilateral agreements high level to avoid complications when there are multiple partners involved in that. The penultimate bullet, of course, is, you know, really, really careful planning. Careful planning, not because of the different geographical locations and the different time zones, but also, you know, the cultures. We have different cultures in the way that we work. And so this has become a very important lesson that we've learned. But finally, but not least, is that so far already, we're seeing the powerful impact of this tripartite collaboration, including, of course, Sean Eakins in the US, is that when you share risks, there is something in it for everybody, then you can also share the rewards. That's what partnerships are about. Each partner brings something to the table. So it's a real pleasure working with Carolina and our other colleagues in Brazil and Russia. Thank you very much. That was the final slide. And I turn back to you, Zuleika.
I'm trying to stop share. Okay. Okay, thank you.
There you go. Okay, thank you very much, Kelly and Carolina, for that very enlightening presentation. It's, you know, Kelly and I go way back with this drug discovery program that was established in South Africa just over a decade ago, I think. It's very exciting to see now how it's contributing together with international partners, you know, on the international stage on trying to solve the new pandemic. And I'm especially excited to see, you know, the tools of the so-called fourth industrial revolution coming to bear to assist us with COVID-19. And I also just wanted to say I appreciate the speakers for sticking to time. So we are still running pretty much on schedule at the moment. I will now hand over to our next speaker. Just before I do that, I just like to send apologies. I'm sure the delegates can see on their programs. We had Professor Nodu scheduled to speak to us today, but unfortunately, because of the travel restrictions that have been unfortunately imposed on us, he could not come back home in time. But we have Professor Hanley Smart, and who will now be presenting to us. She's from the University of Pretoria, and she will be speaking to us about the BRICS ICT Alliance for Smart Resource Utilization to combat global pandemic outbreaks, and partner countries are Brazil, Russia, and India. Over to you, Hanley. Thank you very much.
Awesome. Thank you so much, Zuleika. Thank you for the introduction and for the opportunity to share our project with you today. So let me just make sure that you can see my presentation, please. I just want to make sure that you can see the presentation, please.
Go ahead.
Thank you. Okay, thank you very much. Thank you very much, colleagues. So I would like to share with you the overview of our project in South Africa. It's funded by the Department of Science and Innovation and the South African Medical Research Council. So I'll be representing the team today and just share with you the the status in terms of what we set out to do. So there's 4 aspects that I would like to discuss. I would share some background for you, the thinking behind the project, the way that we've allocated the work across the partners, the collaboration partners, a quick overview to show you some of the artifacts that we've developed and delivered already. And then currently our 3 countries are working in parallel. So I'll show you where the work will come together. So I have high-level feedback from Russia as well as from Brazil. And then I just want to reflect on the collaboration and the status so far. So our project has the 3 primary investigators is from Russia, Professor Vladimir Sudakov. He's from the Kaldish Institute of Applied Mathematics. Professor Esther Colombini is from the University of Campinas. Now, Vladimir is a mathematician. Esther, a strong focus on robotics. And especially application of robotics in health applications. And then I'm from the University of Pretoria in the Information Technology, School of Information Technology, strong focus on modeling, simulation, and systems thinking. So creating a digital twin, if you like, of whatever aspect we are investigating. And this is the way that we've constituted the team. Now, by now, colleagues, you are well familiar with these graphs. Edwin, the first speaker, also highlighted this. And that is that we're continuously seeing these spikes in terms of the pandemic. So from our capability and the capability in our team, our focus was to say, is there any way in which we can be in a position to predict, to use predictive models to predict 2 things, that there may be a spike, and secondly, what do we do? How do we optimize and allocate our resources optimally when we do have a spike in infections? So this formed the basis of our thinking in terms of the project. So the way that we structured the work and the way that we thought about the smart resource utilization was threefold. And the Russian scientists looked at decision making. So what is the impact of management decisions? So in this instance, when we talk about management decisions, we talk about social distancing, wearing a mask, locking down. So any decision that a government or a national authority take in terms of combating the pandemic. The Brazil scientists are looking at predictive models in terms of system correlation, symptoms. So if is there any systems that they can utilize to predict that there may be a spike on its way? And then the South African team focus on the healthcare supply chain. So when we have the management decisions, either to lock down or whatever the decision is, we have a view of a possible increase in infections. How do we optimize our healthcare supply chain in order to optimally distribute our resources? So although the work definition, if you like, or the scope definition of the work then almost form a chain, there is an integration in terms of inputs among the different projects, and I'll illustrate that a little bit later. So what tools are we using to implement our different approaches? From a Russian perspective, Vladimir and his team are focusing on fuzzy logic models. So using multi-agent modeling and multi-criteria decision analysis. So it's artificial AI-based and ML-based models for prediction in order to model and to understand the outcome of management decisions. From the Brazil perspective, also using machine learning models and predictive models, Esther and the team is working with healthcare professionals as well in terms of symptoms specifically. And then from a South African perspective, the tools or the software tools that we will use is simulation simulation model. So I've mentioned that we want to build a digital twin of the supply chain, the healthcare supply chain. And in order to create a digital twin, we need to create a digital model of the healthcare supply chain. Yeah. So once we have the digital model of the healthcare supply chain, we can then do the scenario testing. And that is really where the work of all three teams come together, is when you start to run the scenarios using this digital model. So what is the impact of this management decision? Lock down one province or lock down one region, or you cannot get to a certain region. For example, you can then start to test those scenarios. So the typical software applications that we are using is then allocated across the three primary investigators. So let's quickly look at where we are at the moment. So I want to start with the project, with the SA project team, just to illustrate some of the work that has been done already. And then I'll show you just where the Russian team and where the Brazilian team, where the work will tie in. Currently, we are still working independently. I mean, we meet and we discuss all the outputs together, but the work outputs will only come together in step 5 or so of the 6 steps. So first step was to define the digital twin in terms of the healthcare supply chain. So you can see here the system definition step was the first step. So here we've done 2 things. We looked at what are the components of such a digital twin. And in that analysis, we've also applied another artificial or another machine learning tool called Leximancer. Leximancer is a natural language processing tool. You can see the Leximancer output in the center of the screen. It is those themed circles that shows you the different components that would typically be that form part of a digital twin of a healthcare supply chain. And the analysis is really very powerful, as it gives you total drill down capability, transparency. So I can look at quite a low level of detail. Say if I choose the theme logistics, for example, I can look at what contributed in terms of the logistics aspect of this model. So the first step was to just identify what are these aspects that should be incorporated or designed into the digital twin. And then perhaps just to highlight that this analysis here is forming the basis for our first the South African team's first academic publication, and that is in progress in terms of an academic output. The next step in the process of creating the digital twin is the module formulation. Now, in order to formulate the model, because it is a supply chain, we are working with a reference framework. So the reference framework for supply chains is the SCORE model. And here on the left, you can just see an illustration of that reference framework, the SCORE model. In terms of the model that we are creating, we are looking at SCORE model level 2, which is a level of process flows. There's one process flow illustrated in the center of the screen, and you can see some of our manual work and our workshop work, where we discussed the different aspects of this model, the supply chain model. And this is now applying the aspects that we've identified in step 1. The third step is now to start to look at the data. So data for the digital twin is a key aspect. And here we've acquired our first set of data. So we could purchase healthcare data. I had to just hide some of the columns due to confidentiality aspects, but we've obtained our first set of data, and the data typically contains depots, so you know, import of medication, which is all regulated, a depot structure, how it will be distributed out to then pharmacies, doctors, or depots for undistribution. And the data is required because that forms the basis for us to actually build the model. And that is the step that we are busy with now, which is our 4th step. And this is to actually starting to build the model. Now we are going to build the simulation model in a software simulation tool called Any Logistics. This is a tool that creates a visualization of your supply chain. It takes notes of your customers, your warehouses, your factories. This is in a logistics language. So we've obviously used it in the application of the healthcare supply chain, and we've just started with this. So I'm really looking forward to, at some stage to share actually what that digital twin looks like. Why do we model it? On the left here, you can see we want to get to a point where we can run the scenarios, different scenarios, and in aid of decision support. So that is the ultimate output in terms of creating this model. So the next 3 steps is now future work. And the next step is verification and validation. And this is really where we start to test the model. And just to debug it, we need to see compatibility across the Russian supply chain as well as the Brazilian supply chain. And then all 3 bodies of work will then come together in the next step, which is the experimentation and analysis. And that's the exciting step, because by that time we would have built the model, and now we can start to run the scenarios and to get actual data out of that. And then the final step of the simulation is then to create the documentation. We need to be able to repeat this, right? So we want to create a sustainable model. So we're documenting everything that we do, not just content, but also methodology and process, so that it is repeatable. And that is what we would like to enable. So let's just quickly look at some feedback from the Russian team. They have already developed quite a few models. They've already published their first academic paper on a specific algorithm that they've developed for the prediction of impact of management decisions. So first paper already published, and here's just some illustrations of the artifacts. We share a repository, I'll show you later, called GitHub, where we can share our computer models, and we can have a look at that. In Brazil, Esther and the team have started to consider the healthcare, working with the healthcare professionals, looking particularly at aspects around symptoms in order to include that in their models. Then let me then just close or conclude this presentation by just sharing, reflecting on some of the lessons learned, and just the wonderful opportunity to work together. And I can just reiterate what some of the speakers said earlier. We decided to follow a very formal project management approach, and that is really working well. Because of our focus of repeatability of what we would like to achieve. So documentation is absolutely key, meetings, and to agree the way forward. We've created a WhatsApp group as a separate communication tool just because of the time difference. So it's easy to leave a message and then Esther will pick it up tomorrow, or Vladimir will see it, in the evening, etc. So we've created a WhatsApp group for easy communication, but the source of our documentation is in 2 folders. A Google folder, we've just created a Google collaboration folder, also where we store our documentation, and then all our computer code we keep on GitHub. So I can look at any time at where some progress from the other teams. The time zone difference we've mentioned, the speakers mentioned time zone difference. Language, especially with our Russian colleagues, is quite a challenge. We had the first time when I could look at the models that the Russian team developed, there was a lot of Russian in the code. So we had to discuss the fact that we'll have to use English just as a common language currently in the project team. I liked Kelly's suggestion of a BRICS language. But that was an interesting aspect, just to see this Python code with all the Russian words in. And the team really works well together. We have such a set of diverse skills, and the way that the work is structured sort of in sequence is diverse skills application, but really complementary skills. And the one thing that sort of keeps us focused, and the one thing that is really a focus for every team member, is to make a difference, to make a difference in terms of our contribution in this pandemic. Thank you, colleagues. That's all from my side. Thank you for the opportunity. Zuleka, back to you.
Thank you very much, Hanli. for that fascinating talk. I really look forward to seeing the outcome of this project. Really, really fascinating. And I hear there's a second vote for the BRICS language. Yeah, we are now going to move to our final set of speakers. We are going to hear from Professor Anneke Hesling from Stellenbosch University, who is going to talk about the epidemiological impact and intersection of the COVID-19 and TB pandemics in Brazil, Russia, India, and South Africa. So they're very brave. They're combining all the 4 BRICS countries. Looking forward to hearing from you, Prof. Platform is yours.
Thank you very much. Can you see my screen?
Yes, I can.
Great. And language is no issue. At least we don't have to speak American English for this grant collaboration, which we're used to speaking a lot. And apologies for this. Just put your beautiful, there we go, into perspective. I would actually just like to invite my colleagues who've joined to switch on their cameras briefly so you can see them and they can wave at you. And so Suan, Mohammed, Aneta, Urvashi, and Irina. So later on, when you see the pictures of them on the slide, you'll recognize who they are. And thanks very much for the invitation to present our project. Yes, we feel very brave, but we are conquering due to teamwork. Lots of women in charge, so that helps. As you can see, we're really looking at the epidemiological intersection of COVID-19 and TB. And as you're now well aware, there have been many significant disruptions to TB care globally. And the latest global WHO TB report looks quite dismal. Disruption to healthcare services, reduced access to care due to lockdowns and other reasons, reduced availability of testing, biological disease interactions between the 2 diseases and others, including HIV, mitigation strategies, and then resource allocation. And for the impact on TB, you know, it's multiple, but really epidemiologically looking at a decline in testing, reduced number of people diagnosed, and accessing treatment, and increased mortality. And so for the first time this year, the global TB report actually indicates a massive decrease in case detection and notification, and that's actually across regions, and you can see a massive drop in TB case detection and notification across all the WHO regions. And you're just looking at Africa, and it's really dramatic across all of the regions. And so these are the regions also which include our collaboration. This is just a snapshot of what happened initially in South Africa. And this looks very similar in many other countries and all of our partner countries, and looks at the first lockdowns and what actually happened with access to gene expert molecular testing for rapid diagnosis of TB and RRTB. And you can see a massive decline in numbers, and this is actually not yet recovered. So if you look globally at which are the countries that contribute to the TB notifications and the lack thereof, so actually where the biggest shortfall is India, not surprisingly, given its massive population and case burden. And then you'll see also other BRICS countries, and we have South Africa in there, we have the Russian Federation, and we have Brazil. So all of 4 of our partner countries actually are included in these missing TB cases, which are really quite distressing. And then also, just to, if I haven't convinced you yet, for the first time in decades, we now actually have an increase in estimated TB mortality. And this is also across all of the WHO regions, Africa region, America, Southeast Asia, European region. And you can see that there's an uptick in mortality. And so we're really not on track to meet any of our global TB elimination control targets. So here comes our project. So we called Impact 19 TB. This is our logo. And it's really, the instigator was really Aneta in Brazil. And Irina Felker, who'd actually visited us before, and she's from Novosibirsk in Russia, the Russian Federation, and contacted us to actually collaborate on this project. And then we were really excited to have Brazil, Russia, India, and South Africa come on board. And it's coordinated by the Desmond Tutu TB Center at Stellenbosch University. And here are our 4 project PIs. Mohammed Osman is co-PI here, and our really amazing project manager and lead, Suan Mihan. This is Professor Urvashi Singh, Annette Tramon, and Dr. Irina Felker, Jakob Schwartz, social sciences, Graham Hodinat. We have a modeler, an epidemiologist, Florian Marx, and very active social and civil society engagement with ASIO leading that in Brazil. So we have 5 interlinked objectives that I'm going to talk through them briefly and look at some updates we have for you. And these are all complementary and intersecting, and some of these are addressed by all of the partners, and some are actually led and coordinated by some of the partners. So the first objective is really looking at the tuberculosis care cascade and the gaps to address and to actually quantify the gaps between steps in the TB care cascade that is attributable to COVID-19. From actually having TB, accessing care, getting diagnosed, getting registered, starting treatment, and having a good outcome. And so specifically, we are addressing gaps 2, 3, and 4. So when you've gone to services, but you have been diagnosed or not, started on treatments and treatment outcome. And we all know that the impact is likely to be negative, but it's actually important to quantify and in order to be able to recover. And so here we're looking at routine programmatic data from public services in all 4 countries and using routine data sources, which come with its joys and its challenges, both for treatment as well as for lab. A joint agreed upon database with indicators across all 4 countries. And then we're looking very specifically at subgroups of interest, including people living with HIV, children, adolescents, elderly people, and people living with drug-resistant TB. We're also looking at TB preventive therapy and not just the people with active TB disease. And the progress has actually been substantial given the challenges of accessing routine data and large national routine data sets. We've developed a minimal data set with indicators across all 4 countries. And we'll be able to look at additional disaggregation via WHO age bands and weight bands for 3 of the 4 countries, which is great. To really enable us to look at those additional populations. We have finalized a data transfer and sharing agreement for this combined 4 country data set. We are awaiting the South African data, but it's been approved. Russian analysis is already underway. Our Russian partners have been amazing, very, very dynamic. And Brazil and India are waiting for the Ministry of Health to release the data sets. And this is a potential challenge for us, but we continue working. We all have very good collaboration with TB programs. And actually in India, our partner there, Professor Urvashi Singh, has specifically formally involved the National TB Control Program, which I think has really been strategic. And just to show you, these are some of the indicators we look at from TB treatment registers, from sputum registers, from preventive therapy, from TB HIV indicators, and then also we're looking at COVID-19 vaccination. Okay, so that's objective 1. Objective 2 is, so objective 1 was actually a joint collaboration with all 4 project partners, and objective 2 is really led by Russia and India. And they are really looking at the impact of COVID-19 on trends in mycobacterial population structure. And in Russia, they've already collected a substantial number of samples. Analysis is really underway with some very early data that I'll show you, and I'll ask Irina to comment on that. In India, is very efficiently using the National TB Prevalence Survey to collect the isolates with the National Geographic coverage of India, samples collected at the community level, and this data transfer is actually in progress. So Russia, as I said, has already made substantial progress here. This is a 2-year project only, so we're really watching the timelines very carefully. And they've collected the clinical database on patients with TB and COVID. And then they've really actually genotyped 192 mycobacterial isolates. And what's very interesting from very early data, they actually show that the non-Beijing genotype, and Beijing is usually thought to be quite virulent. We'll have a lot of that in the Western Cape, actually, where I work. We're more prevalent in people with TB and COVID, which really poses the question of virulence and what it needs to actually make someone ill with TB and COVID concurrently. So this is very early, and the group is actually looking at publishing this data quite soon. The third objective is to determine the effect of TB on patient-level COVID-19 outcomes. And this is really looking at TB with and without COVID. And this is a Brazilian and Indian partnership primarily, with support and academic and scientific input and analytic input from the rest of the group. And Brazil has a very strong collaboration with the national TB programs as well as other partners. The data linkage process, which is quite a unique skill and quite important with these large routine data sets, has been finalized. Aneta can comment on that. And rapid sharing of data by the Ministry of Health is required there to move this forward. And India is actually using their national ICR COVID database and their national TB database, NICSHE, and they already have identified 5,000 patients with both TB and COVID-19. And from the early data, they can observe a higher case fatality in TB patients with COVID-19 compared to those with TB only, which is not surprising. And that's also what's been observed in South Africa with the early data from the Western Cape. And this is from Aneta group, and this looks at the process of data linkages and looking at the sources of surveillance. And you can see multiple sources of data to bring in the data and link the data for COVID patients and COVID TB co-infected patients, and linking these data to be able to have a joint single database with multiple sources of surveillance tapped into that. Objective 4 is really to determine the programmatic and the country responses to mitigate these negative impacts of COVID-19 on TB services. And there are two components. It's looking at healthcare providers, and then it's looking at patients and caregivers. And it's important to look at caregivers because there are children or elderly people that might be frail. And so there's a healthcare workers survey that's self-administered, and this is like a consumer satisfaction type survey in all 4 countries again. And this instrument was really jointly developed by all partners and with a lot of input by community advisory structures and community advisory boards. It was piloted in Russia and then actually started implementation in June. And so you can see we have a nice cover in Brazil, in Russia, a national level in India, and then in South Africa, 3 high burden provinces with very different resources. And we've already actually completed the survey in more than 290 healthcare workers. We actually have not had particular challenges with languages because we feel we all communicate well to each other in English, but we have to translate into multiple languages to make this accessible to healthcare workers and our participants and patients in the field. And then we're doing in-depth qualitative work, and this is more targeted, and this is in Brazil, India, and South Africa, to get the more rich experiences of healthcare workers in the field, and that is just underway now. And there's a very high-level profile of how many healthcare provider surveys have been collected to date. And I wanted to just briefly share with you some initial experiences from the Russian team. Looking at healthcare services overall, you can see there's been a massive negative impact on vaccination, which mostly affects kids, and also significant impact on TB. And most of that TB impact seems to be on presumptive TB patients, so people who need to be tested for TB. Then component 2 actually looks at people affected by TB themselves, the patients, and it's looking again at the same process of looking at a patient and caregiver survey, which is relatively high level. And then again, in-depth qualitative interviews with patients and caregivers in again, the same 3 countries, and using the same processes to co-develop instruments, pilot them, and then again, jointly share the experience and the analysis. And this work is also going really well with the data collection already underway, and again, quite iterative analysis. So you'll see that there's focusing on the healthcare services and healthcare workers as well as the patients, looking at quantitative data approaches as well as more in-depth qualitative work, and these are really complementary. And from the patient and caregiver surveys, this is a breakdown of how many we've collected to date, which is quite nice, and a nice distribution between the countries. And then again, preliminary data from the TB patient survey from Russia, here again showing that experience of TB testing was actually better before COVID, based on this initial survey results, than it was after the onset of COVID. Finally, objective 5 is looking at the impact of COVID and response measures by using mathematical modeling. This is really led by South Africa and Brazil, but again, with input from all of our other colleagues. And close collaboration with the CIMA at Stellenbosch. And this is using existing developed calibrated transmission dynamic TB models for both countries. And then actually models the impact of the response measures on TB incidence and mortality, but also specifically models the impact of recovery measures on the TB care cascade. And so when we're doing these interviews and collecting the data, we're not only looking at all the bad, but actually what has worked to mitigate. And so what strategies, for example, have worked. Can you test people in different places? Can you test for TB and COVID at the same time? Can you take people their medicines home? Can you dispense it in a different way? So also how to recover and how to sustainably recover and actually model these strategies. And this is in close collaboration with the South African TB think tank. And again, it's a strong focus throughout our project, but especially here also on capacity development and exchange, and really excited about having a comparison between the Brazilian and South African national TB model. For collaboration and capacity building, we really have strong regular communication, and it's really been a great collaboration with regular meetings. The data sharing agreement, getting that finalized has been really key. I think a potential challenge, but also an opportunity, is the Department of Health involvement and releasing data and getting access to routine data, which is notoriously challenging. We really have a strong focus on capacity building, and we would like to expand this going forward with several postgraduate students, planned in-person exchange, but perhaps less so in the context of our new variants. And we're planning a joint first big project meeting in early next year, which will probably have to be virtual. We also just wanted to highlight the importance to us of community and civil society engagement. It's not just about the science, but also about engaging people who have the answers to help us and think about how to interpret findings, how to solicit input, and how to disseminate our results. So our Brazilian colleagues have been incredibly dynamic, and we had our first civil society webinar already in September, and we had more than 105 people, and you can see the very strong participation by all 4 our project countries. And this has really been excellent, and was regarded as, had very positive feedback. Project milestones look good, considering that we've only been going for a couple of months, but it's a short period, and that's one of our big challenges, is to have the impact that we want to within the short grant funding period. We have several papers underway, a review article on TB COVID-19 led by Aneta will be submitted shortly, and then the initial survey work for healthcare workers and patients, which will also be available first. So we really wanted to thank you for funding us and supporting us. Thank you for the great collaboration. And you'll see that there are a multitude of collaborators just in South Africa, and these are formal total project partnerships and many more in country that keep on growing. So thank you very much.
Thank you very much, Annika. That's a massive project, and such an exciting project and an important project, considering the burden of TB in all of these countries. And it's really good to see such an important disease not being left behind, with all the focus on COVID-19. That was, thank you very much for the presentation. And I would just like to remind our participants that you can send through your questions. You can use the QR code to send your questions and comments to engage with the panel. And I'd just like to thank all the panel members for using the time very, very efficiently. So we've got a little bit of extra time for questions. And I'm just going to kick off the question sessions whilst we wait for the questions from the panel members. My first question is to Edwin, and we just wanted to find out, your presentation understandably focused a lot on the new variant Omicron, but we just wanted to find out, is the team in SA as well as your partners also focusing on the Omicron variant, and what are their experiences? And also, if you can, in your answer, just elaborate how how the partnership is working with the other collaborators.
Correct. So we've been having regular meetings since the past 4 months. It's been a bit of a slow start, but since we wrote the initial publication, and given the fast pace of genomic surveillance in the past 12 months, most of the objectives of the original application has actually already been completed. So we are currently working on our first collaborative publication, looking at the various responses to the pandemic in the 5 countries, as well as the epidemiological profile and molecular profile. That should come out early in 2022. So yeah, we continue to engage with our partners in the other 4 countries. and it's definitely a growing and very active collaboration. Since the identification last week, we've already been in contact with our partners in Russia and in China and in Brazil as well. So things are definitely moving very quickly. Thank you.
Okay. Thank you. If I could just ask a follow-up question, perhaps it didn't come through to me and perhaps other people as well. What are the different roles that the partners played or you envisage them playing going forward in the project?
So it's mostly everybody sort of have the same skill level on all 5 countries. So it's mostly sharing analytical pipelines.
Sorry, Edwin, can I ask you to switch on your camera, please, if you don't mind?
Sure. Can everybody see me? My internet is a little bit slow.
Yes, thank you.
Okay.
Oh,
So
okay, that's it. Yeah.
It mostly involves sharing of analytical pipelines and also infectious agents. So our colleagues in China and in Russia has asked us whether or not we could share cell cultures of the neo-Omicron variant. It has already been raised by some of our colleagues in the call that it's quite difficult to create material transfer agreements on such short notice. So there's definitely also obstacles to collaboration that we can discuss. Thank you.
Thank you very much. And I next have a question for Kelly. Is your project focusing on any particular variant, and has the the identification of Omicron had any impact on your project plan?
No. So the straight answer is no. However, when we get to the point of testing for antiviral activity on the different cell line, that's when we begin to profile compounds on multiple strands. So this is a very typical thing. For example, if you're working on TB, you do the initial screening on, let's say, the H737RV, which is a lab adapted strain. But when you find something interesting that you've optimized that's moving forward towards the clinic, at that point, then you test on different strands to see how that particular drug responds to various strains, whether they are drug resistant or sensitive. So with COVID, in a nutshell, we're going to get to a point in the program where we then want to know how those compounds that we're identifying from the AI, from the artificial intelligence, are working on multiple strains. But until then, we have to focus on one thing first and then begin to expand. Thank you.
Thanks very much, Kelly. And I just wanted to ask Hanli, who do you consult? I mean, at the first part, of your process, the system definition. I imagine that must be very important because that's the input side. And how do you handle the consultation? Who do you consult for that?
Right. So we consulted, I guess, 3 aspects. The first was the literature, so the body of knowledge in academic literature around modeling and creating digital twins, particularly of supply chain. So secondly, we are very fortunate to have access to experts. So we have access to supply chain experts, both here in South Africa and with our partners, that and not just supply chain experts, but also healthcare supply chain experts that is in the industry, that could explain and sit with us. And some of those images that I've shown are some of the drawings that was done while they were assisting us in just creating that systems view of the model. And then the other is data. So I guess for us, there's two aspects. The one is a top-down type approach, and that is to use the experts and to use the literature, but there's also the story that data tells, right? And if I have the data with geolocations and I have the warehouse, I've got the depot, I've got the doctor, the pharmacy, the hospital, when you import that into the simulation, it actually creates a model for you. So the data also tells a story. That's the point that I just wanted to make. So those are sort of the three inputs. Thank you.
I will unmute myself. Thank you very much. I just want to remind the participants, you can send through your questions using the QR code. I just wanted to, I just have a question now for Anneke. Would you say that the mortality in TB patients has been in any way linked to the different variants? Have you seen any variation?
Yeah, thanks, Aleka. That's a really interesting question. Yeah, so I think we don't have the answer yet, although, looking at, we will have to look at more data over time. And thankfully, we can do that relatively long term with this project because we're looking at data historically before COVID was introduced into South Africa and the other 3 countries, and looking at trends longitudinally. So we will then be able to import other sources of data like the emergence of variants, etc., and control for that in the analysis. And I think with the modeling, we're able to allow for some of that. But I think there are also many confounders epidemiologically. And in South Africa, major confounders are very high HIV prevalence, which might drive poor outcomes in people with TB as well as variants and poor outcomes in general. But I think the long answer, or the short answer to your question is really that we might be able to tease that out over time, but I think we also need to consider that there are many confounders. And also, even if you look at the mitigation strategies and the restrictions, etc., that these vary considerably over time as the variants also vary, as vaccines and uptake vary. So we can certainly look at that in the analysis, but we don't have any data to suggest that yet. And I'm not sure that we'll have a definitive answer, but that's definitely an area we will be looking at. Thanks.
Okay, thank you very much. I'm trying to see if we have any further questions in the chat room. Okay.
So, Leka, I'm not sure if any of my colleagues from Brazil or Russia or India wanted to comment on that question.
Certainly, we can give them an opportunity.
Yeah, I agree with Anneke. That's very difficult to attribute or even to try to correlate the variance with the mortality. I believe there's, as Professor Haskin just told us, there is a high interference of vaccination policies and mitigation policies that will interfere with the mortality in TB. And unfortunately, we believe that the very sharp decrease in TB detection that she showed us today will unfortunately result in higher rates of death in the future, still higher than what has been shown in 2020, unfortunately.
Thank you very much, Anita. Arina, do you want to comment? Sorry, Arina, do you also want to comment?
Irina's on mute.
Sorry, dear colleagues, can you hear me now?
Yes.
I'm very sorry. I have some problems with the connection all the time when I want to say something. So in Russia, we also cannot say on this stage if we have any relations between the strains of the type of virus and their mortality rate. But I could say that we definitely registered the high mortality rate among patients with TB because of COVID-19 in comparison with the general population. So let's see, maybe their next few months will give us some answer regarding their any relations between the strains of Mycobacteria tuberculosis and virus.
Hey, thank you very much. And then I have a question for, we still have a couple of minutes for all the panel members, and that relates to capacity building. I remember, Anneke, you touched a little bit on this, but I just wanted to ask how, I understand the time constraints, this is COVID-19, a lot of the projects that are focused on, trying to find solutions that will help with the pandemic in the very near term. But I just wanted to find out, have you found opportunities to incorporate a capacity development element in your projects, and how have you factored, the timeframes into your planning? Okay, I will give whoever wants to take it first.
Maybe
okay.
Add to that, so since we've already touched on that, I think all the countries involved in our project are extremely committed to capacity building, not only academic capacity building, but also capacity building within healthcare services. And so actually to also use, there's a lot of routine data being used in our project, and to use the routine data as a training and capacity building opportunity for partners working in healthcare services is also really important to us, so that the value of utilization of routine data can be seen and can be capitalized on. I think since there's a lot of data that's being collected, there's some new data collection, but there's also quite a lot of existing data that's being utilized. And so there's really a huge opportunity for postgraduate students to become involved, even if it's only for example, partial support of a PhD. And also we have found in our respective countries that there are many other partners who are interested in collaborating and further sharing data. And so, for example, Anita can comment, in Brazil, there are two PhD students who will be working on the same data set, with two collaborators coming together there, including our project data sets, to work on it together and carving out who's going to be doing what, to just and just recognizing how limited the time frame is and actually best supporting students. There's also some project support through funding that can actually support junior researchers, for example, who plan on doing their PhD to actually be salaried while they do it. So 2 years is fine for a master's degree. For a PhD, it's a bit short, but there's an opportunity for this project to contribute to postgraduate work, and also for us to actually continue this collaboration beyond the project, which I think is also really important to us.
Thank you. And Anneke, if I can we just have about a minute left, if I could give it to Handi.
Thank you very much. Yes, thank you very much. We also have a strong focus on capacity building from 3 perspectives. Already we have master's and PhD candidates, as well as postdocs working on the project. Secondly, in our work plan, we've actually built in learning and development stream. So not just for the students, but also among the team. So what we can learn from Brazil, what we can learn from Russia in terms of the different skills, also the different software tools that we use. And then the third component is into next year, when we actually have the model, is to then also engage with communities and then to take it further, because once we have it, we can reuse it.
Okay, thank you very much. Edwin?
Sorry, I was muted there. Yes, so in our last NGS BRICS call with our colleagues, we've discussed the possibility of doing a webinar training series early in 2022. Us ourselves here at Siri and Chris, did a 2-week training course, hands-on, in collaboration with the Africa CDC, training 17 different African countries, people from national health institutes, how to not just do the sequencing, but how to do the analysis, and then how to analyze the data and report it back to a Ministry of Health. So yeah, we've been very active in that.
Okay, I thank you very much. I do not see any further questions. I'd just like to take this time to thank all our panel members. This has been a fascinating presentation, and it's really great to see how much progress all these projects have already delivered in such a time frame. And I wish you all the best, in continuing your project. But thank you very much, everybody. This is fantastic.
Thank you. Thanks
Cheers.
for joining.
Bye. Bye, Donkey.
Thank you. Bye-bye.
Bye. Bye-bye.
Thank you.